Good evening, Soumya Jyoti. Good evening, everyone. How are you all? Are you all good? Okay, did you complete your assignments? What about assignment 3? There are still students who haven't submitted their assignment 3. Okay, so application is still open for assignment 3. You can apply, you can submit your assignment, okay? Okay, so during class, if anyone wants to ask any question, you may raise your hands, okay? So that way I will know that you want to ask something. Is it clear? Okay, let's continue with our today's session. Okay, so last week we learned two important sequence alignment algorithms, Needleman-Wunsch and Smith-Waterman. And we learned that these algorithms can accurately compare the biological sequences. But there is a problem. So the problem is, suppose I have this DNA sequence. Okay, this is my DNA sequence. And I want to know that which gene is this, or which organism does it belong to, or does a similar sequence already exist in the database. So I cannot compare my sequence manually with every sequence which is available in the NCBI. As we know that NCBI contains millions of sequences, so we cannot manually compare our sequence with all the millions of sequences present in the NCBI. Correct? We cannot do this task manually. And another problem that we have is we can use the Smith-Waterman. We already understand the Smith-Waterman algorithm. So we can use the Smith-Waterman to compare our sequence with every sequence in the NCBI, but it would be extremely slow. So there is a time problem and effort problem as well. So scientists, they needed a faster method. Scientists wanted a tool that could search huge databases. As we already know, all the databases, they contain millions of sequences. So scientists wanted a tool that could search these huge databases. It could find the similar sequences and it could produce the results very quickly. So to solve this problem, BLAST was developed. Now, what is this BLAST? So, BLAST stands for Basic Local Alignment Search Tool. So, BLAST is used to search the biological databases for sequences similar to our sequence. Okay, I'm repeating it again that BLAST is used to search the biological databases for sequences which is similar to our sequence. Now, what does BLAST do? So for example, this is our sequence here, okay? So BLAST, it will take our sequence and it will search all the databases, it will search the large databases just like NCBI to find the similar sequence to our sequence. For example, let's take an example to understand this. So suppose a researcher, there is a researcher and he discovers a new DNA sequence. So now the first question is that have scientists seen this sequence before? So instead of comparing it manually against millions of sequences in the database, they will simply run BLAST. So BLAST, it searches the database and it reports back similar sequences. Now, why BLAST is fast? We are talking about that BLAST is very fast. Now, why is this fast? So this is the key concept of this entire lecture, that why BLAST is fast. So we already know that Needleman Wunsch and Smith Waterman, they try to examine the alignments very carefully. Correct? Whether it's Needleman Wunsch or whether it's Smith Waterman, they align the sequences very carefully. Correct? But BLAST here, it uses a shortcut. So instead of comparing the entire sequence immediately, as we know that Smith, Waterman and Needleman Wunsch, they compare the entire sequence, whether it's local alignment, whether it's global alignment, the entire sequence is matched, right? There is a complete alignment. So BLAST here, it uses a shortcut. And what is the shortcut? That instead of comparing the entire sequence immediately, BLAST first searches for short exact matches. And these short exact matches are called word. What are they called? They are called word. For example, this is our sequence again. So what will BLAST do? BLAST will break this sequence into small words. So here, for example, this first small word is ATG, then it is TCG, then it is GCT. In the same way, CTA, TAG, here we have AGC, we can see AGC, another small word, and the last small word CTA. So BLAST here, it breaks our sequence into these small words, and then it searches the database for these words. Now the important thing to understand here is that BLAST does not align everything from the beginning. Okay, just as we are talking that BLAST uses a shortcut and this is a shortcut. So BLAST, it does not align everything from the beginning. It first finds these small matching regions in the database, all the sequences which are available in the database. So BLAST, it first finds these small matching regions from our sequence, and only then it performs a more detailed alignment around these regions. Okay, so this makes BLAST much faster than the Smith-Waterman algorithm. As we know that the Smith-Waterman algorithm, it performs the local alignment. BLAST here also performs the local alignment, but it performs fast local alignment. And here we got the idea how BLAST is fast. Okay, till here, do we have any questions or is it clear? Is there any confusion till here? Any confusion, everyone? Is it clear about BLAST? Okay, Akshari, very good. What about other students? Okay, very good. So let's continue. So first of all, let's understand what happens inside BLAST after we submit a sequence. Okay. So here, just assume that this is our sequence. So the user is us, right? The user is you. So the user, he or she provides a sequence to the BLAST. So this is called query sequence. Okay. This is called query sequence. Just like we gave the input of our sequence in the Smith-Waterman and the Needleman-Wunsch, the emboss tool, there we gave the input. In the same way, we give this input, but here we are calling it a query sequence. Okay, so the user provides a query sequence to the BLAST. Now, as we have discussed that BLAST, it does not immediately compare this whole sequence. Instead, it breaks the sequence into these small pieces which are called word. Now, for example, here the word size is 3. What is the word size? It is 3. We can count A, T, G means 1, 2, 3. So here the word size is 3. So these small words, they will be searched in the database. Now the question is that why does BLAST do this? Why is BLAST breaking our sequence into the word size of 3? So why does BLAST do this? So for example, let's understand this with an example again. So imagine that you are searching for your friend in a huge city. Okay, you don't know the information of your friend, you are just searching for your friend in a huge city. Now, instead of checking every person carefully, what you will first do, you will first look for people who roughly match the description of your friend. For example, your friend's hair color is red. So obviously you will look for those people first whose hair color is red, right? Similarly, BLAST here, it first searches for these small matching words and then it does the complete alignment. Okay, so now for example, the first word is ATG. So for example, that this first word ATG, it is found in 4 sequences in the database. In the database, we have millions of sequences. So in the database, this word ATG is found in 4 sequences. Let's name the sequence, sequence 1, another sequence, sequence 5, sequence 20, and sequence, for example, 105. Okay, so we have these 4 sequences, sequence 1, sequence 5, sequence 20, and sequence 105 from the database. And our ATG here from this sequence, it matches those 4 sequences in the database. So these sequences from this database, the sequence 1, the sequence 5, the sequence 20, and the sequence 105. So these become potential candidates. Are we understanding the term that why these sequences become potential candidates? Because our word ATG matched in those sequences. Okay, now when BLAST finds a matching region, so here we just discussed these 4 sequences, that ATG word here was found in those 4 sequences. So when BLAST finds a matching word in the database, it is called a hit. Are we understanding the term hit? That when the BLAST finds a matching word from our query sequence in the database sequence, it is called a hit. Okay, so this example here, this is a hit. Now again, the important point to remember here is that at this stage, okay, at this stage, BLAST has not aligned the full sequence. Okay, here from our query sequence, we have this word ATG. From the database sequences, for example, all those 4 sequences, ATG matched with our query sequence ATG. So this is called a hit. Here we are not aligning the complete sequence. So at this stage, BLAST has not aligned the full sequence. It has only found the small matching regions. So this is where our next step in BLAST is called extension. Now, in this extension, suppose BLAST finds the ATG in the database. So instead of stopping there, the BLAST found ATG from our query sequence and from the database sequence. So BLAST will not just stop here. Instead, it will extend the match in both the directions. So here we can see that ATG, after the ATG, our sequence is this and the database sequence is this. So what is BLAST doing? It will search for this direction as well, and it will search for this direction as well. So BLAST here, it extends around the hit to see whether a longer alignment can be found or not. Now, why are we doing this extension? Because a short match of 3 letters, it can be happen by chance, right? ATG can be present in any sequence. For here, we are only taking a small example of ATG. That's why we can understand that ATG sequence here, it can be available in many sequences. But the sequence, our query sequence, if it matches with another sequence in the database, so here BLAST sees that what are the chances that our query sequence is matching with the sequence in the database. Okay, again I'm repeating it for you that here we have this 3 short letters ATG. So here it is a short match. So it can happen by chance. It has a very common reason that it can happen by chance. But this longer match, is it biologically meaningful, or is it again a chance? It happened again by chance, just like this. Okay, so BLAST does this extension steps. So here we have the ATG. So BLAST will search for after the G and here before the A. We can see this arrow and it will match our query sequence with the database sequence to see if there is a longer alignment or not. So are we clear with the extension step? You can raise your hands if you want to ask any question. Otherwise, I will just continue. Okay, so let's continue. Here we have understood till the step extension. Now, after extension, the next step is alignment and scoring. So we already understand alignment and scoring from the previous algorithms. So here, after extension, BLAST performs the alignment here and it calculates the score. So as we can see, A and A is a match. So a score will be given. T and T is a match. A score will be given. Here we can see let me see here, A and T is not a match. So there is some error here in this picture. So we can see A and T is not a match. So we see a dot there, and for mismatch, the score will be given. And we already understand the higher the score is, the better is the similarity, the better is the alignment. Now, after the alignment and the scoring, we got all the scores. Correct? We got all the scores at what is the highest score, what is the lowest score. But after this step, BLAST here, it asks that is this match biologically significant? Means, is this match biologically meaningful, or again did it occur just by chance? Just like here, ATG happened again just by chance in many sequences from the databases. In the same way, is this match here is biologically meaningful, or did it happen by chance? So to answer this question, BLAST calculates the E-value. Now, E-value here is one of the most important values in BLAST. So first of all, let me know if you have any confusion how the BLAST works. Only after that we will continue with E-value. Are we clear how the BLAST works? Everyone? Okay, so let's continue with E-value. So, E-value here, it is called expectation value. Okay, now this E-value, this expectation value, it tells us that how many matches, okay, listen to this very carefully, that how many matches with a similar score would be expected to occur by chance in the database. Okay, can we understand this line, that what is the meaning of E-value? Means how many matches. These are all the message, our query sequence, and this is the sequence from the database. In the same way, we can have 4, 5, 6 sequences aligned with our query sequence. Okay, this is our one query sequence, and for example, we got 10 sequences which are aligned with our query sequence. So the E-value is saying that how many matches here, and they have all the scores, right? This is sequence. For example, let's name our sequence, sequence A, and let's name this database sequence, sequence 1. So sequence A versus sequence 1, sequence A versus sequence 2, sequence A versus sequence 3, and so on. Okay, so here we have so many matches. So how many matches with a similar score would be expected to occur by chance? So again, is this happen by chance just like this here, or is it have a biological meaning? So suppose that BLAST, it gives us a result with E-value 10. Okay, again let's come back to this example here. For example, this match here, BLAST gives us the E-value of this match 10. Now the E-value of this match is 10. So this means that we expect about 10 matches of this quality to occur simply by chance. Okay, I'm repeating it again. Here, if the E-value of this match is 10, so this means that we expect expect about 10 matches of this quality to occur simply by chance. I know you didn't get it properly, but we will learn it in deep. Okay, so here the E value is 10. Now this is not very convincing. Okay, E value is 10. This is a very high number, means the chance is very high that it expected just by chance, just like this. So here E value is very high, so it is not very convincing. Now suppose the E value is 0.01. For the same match, the E value is now 0.01. So this means only 0.01 such matches are expected by chance. So are we understanding the difference between 10 and 0.01? Means here the E value is 10, means the chance is very high. Here the E value is very low, means chance is very low. So here if this match, the E value of this match is 0.01, that means these matches are expected by chance very low. Now, for example, E value is this. Now, what is the meaning of this? Here, 1e minus 5. So, it means that 1 into 10 raised to power minus 50. 1 into 10 raised to power minus 50. That means we have 50 zeros before 1. Here we have 1 and before 1 we have 50 zeros. So, that means again this is a much smaller value. Right? We are understanding 10 is a bigger value, 0.01 is a smaller value compared to 10, and here 1 into 10 raised to power minus 50 means 50 zeros before 1. Now it is a very extremely small value. So this means if the E value of a match is this, so this means that the probability of that match occurring by chance is extremely low. Okay, let me repeat it again, that if the E value is this, that means the probability of this match occurring by chance is extremely low. So this is a very significant match. Now, what is the rule? So easy rule to remember: lower the E value, more significant match, means the match that is to be happen by chance is extremely low, and higher the E value means chances are very high that this match happened by chance. Are we clear with E value, or do we have confusion with E value? Is it clear? Any question related to E value? Because as a beginners, when first time we learn about E value, we tend to confuse for between the higher value and the lower value. So you have to take very good here. Here you have to observe something very good, that this is the rule: lower the E value, more significant the match, means less chances that the match happened by chance, and higher the E value means less significant match, means that match happened by chance. Okay, now what are we going to do? We are going to open the blast now. Okay, very good Soumya, very good Akshari, very good Shraddha, very good Siddhesh, very good everyone. Now we are going to open the blast. So the blast tool, it is available within the NCBI. So either we can search for NCBI or we can directly write the blast here as well. Okay, you can do anything that you like. But first of all, let me show you in the NCBI where is blast. So you can directly open blast from here as well. But let's open NCBI. So here is the blast. Okay, this is the blast tool. So let's click on it. Now, this is the blast basic local alignment search tool. So before we perform the blast search, we need to understand something important here. Here we can see 4 different blast programs. Here is nucleotide blast, here is protein blast, here is blast X, and here is T blast N. So the question is that which blast program should we use among these 4? So the answer, it depends on what type of sequence we have. Okay, what type of query sequence we have. So here, first of all, okay, this is the first thing, that what type of query sequence we have, and the another thing is which type of database that we want to search. Here we understood that what BLAST does, it searches against the database, and we already understand that there are many types of databases, correct? So that is what we are seeing here, the nucleotide BLAST, and here the protein BLAST. Means if our sequence, our query sequence is DNA, so among which databases we want to compare our DNA sequences with. Okay, so let's understand it. So first of all, we have the nucleotide blast. So this nucleotide blast here, it is used when our sequence is a DNA sequence. Okay, our query is a DNA sequence. That is where we will be using the nucleotide blast. And second thing, that if we want to compare our DNA sequence with the DNA database, okay, our DNA sequence to compare in the DNA database. So these are the two important things. What is the sequence and which database we want to compare with. Okay, so here we will use the nucleotide blast, and it is also called as blast N. So N stands for nucleotide. And here we have the protein blast. So protein blast is used when our query sequence is a protein sequence and we want to compare it with the protein database. So that is where we will be using the protein blast, and it is also called blast P. So P stands for protein. Okay, and here we have blast X. So this blast X is used when our query sequence is a DNA sequence. Here we can see nucleotide is written. So there is no need for any confusion. What is blast X and what is T blast N? Here nucleotide and protein are clearly written. So we have no confusion in these two. But here, to not have any confusion, you can read this line. Okay. So this blast X is used when we have a DNA sequence and we want to compare it with the protein database. So here what BLAST does, BLAST first translate the DNA sequence into protein sequence. Means we already know DNA sequences have ATGC and proteins have amino acids. So we cannot just simply compare DNA and protein. So that's why BLAST here, it first translate the DNA sequence into protein and then it compare with the protein database. So are we clear with BLAST X that if we have a DNA sequence and we want to compare it in the protein database, that is where we will be using BLAST X. And the another BLAST program is T-BLAST N. And here we can already see if our query sequence is a protein sequence and we want to compare it in the DNA database. Here translated nucleotide is already written. So nucleotide means DNA, translated means DNA are being converted into protein sequences. So here, if our query sequence is a protein sequence and we want to compare it against the DNA database, so here we will be using the T-BLAST-N. So here the DNA sequences, of the databases, they are translated into proteins so that it can compare properly. Okay, so these are the first BLAST programs that now we understand when to use these programs. So firstly, we will be using the nucleotide BLAST program. So let's open it. Okay, so here we can see above blast N, blast P. Blast N means nucleotide blast. We have already selected this. Blast P yani ki, means protein blast. Here we have blast X. Here we have T blast N, and here we have T blast X. Okay, so here we can either enter the accession number or we can either enter our sequences, just like we pasted the sequences in the Smith-Waterman algorithm in those previous two algorithms. In the same way, here either we can enter the accession number or we can paste our sequence or we can choose the file. So we already have a file, we have human insulin file and rat insulin file that we already downloaded. So we will just click on choose file and this was our human insulin file. Now let's click on open. So this was my file name sequence1.fasta. So I have uploaded my file. So here the job title is the same as the emboss tool. Here you can enter your job title if you want, and here you can see database is already selected, and here all you just have to do, you just have to submit your file. That's it. And one more thing that I can show you here, you can see the word size. We just discussed about the word size. So here you can see that the word size is 28. That means from my file here I have DNA sequences. So what BLAST is going to do, it is going to divide it into 28 word size, and then it will compare it in the nucleotide database. Okay, so here you just have to submit your file here, or you can paste the accession number, or you can write the FASTA sequence, and you just have to click on BLAST. Now let's wait for 2 minutes. Now this is our result page. Okay, so here you can see that the job title was this was my file name, Homo sapiens insulin mRNA. Right here we have the program that we used, BLAST, and the database that it was compared with our file. It is the nucleotide BLAST. There is the description, molecule type was DNA, and the query length is 333. Means the DNA sequence that I had in my file was 333. So now let's scroll down. Here, what are we seeing? Here, these are all the sequences from the nucleotide database that has been aligned with our query sequence. So can we see how many sequences aligned with our query sequence? So these are all the sequences, okay? And here, we are seeing some sections here. So first of all, let's talk about the query cover section. So here you can see that in the query cover section, it is 100%. Here it's 100, here it is 99%, here 100%. So mostly it is 100%, here we have 86%. So again, mostly it's 100%. Means the query cover is 100%. Now what is the meaning of query cover? So it means this query cover means query coverage. So it tells us that how much of our query sequence participated in the alignment. That means our query sequence length is 333. So here 100% is written. That means from position 1 to position 333 means from starting till the end, our query sequence participated completely in this alignment with this sequence in the database. And here, as we can see, we have 99%, and here we had 186% as well. Here is 86%. So here that means our query sequence participated only 86% in the alignment with this sequence from the database. So are we clear with the query coverage? What is the meaning of the query coverage? And second, here you can see percentage identity. So this section here is also showing us 100%. Now this percentage identity, it tells us that out of the aligned positions, here we can see 100% means from position 1 to position 133. So here percentage identity means out of the aligned positions, how many are exactly identical. For example, our query sequence is ATGC. Okay, and this sequence is also ATGC. That means the percentage identity is 100%. All the position matched here. Now, for example, our query sequence is ATGC, and this sequence is ATGT. Okay, I'm saying it again. Our query sequence is ATGC, and this sequence is ATGT. Now, here only 3 position match. Correct. Now, here the percentage identity will be 75%. Okay, so this percentage identity, it tells us that how similar the aligned sequences are. Are we clear with query coverage and percentage identity? Do we have any question between these two? Okay, so here we have E value. So we already understand what is E value, and we can see E here. And here E means that there are 171 zeros before 1. So it is 0.000 and so on. After 171 zeros, we have 1. So here it is a very small value, very small value. Okay, so here we have E value 1E 171. So here you can see 4E 171 means 171 zeros before 4, and here 170 zeros before 2. So now all of these E values, they are very small. So lower the E value, better is the alignment. Okay, so all of these matches, all of these sequences that align with our query sequence, all of these are extremely significant match. And here we have maximum score. So we already understand the maximum score. If one position has a match with another position, means from our sequence position 1 and from this sequence position 1 we have A, so we give it a score. For match we give a score, for mismatch we give a score, and for gap we give a score. So now the maximum score is 616. So this is the highest score, 616. Here we can see we have score 614, here we have 612, here 608. Now we can see that as we are going down, the score is decreasing. Okay, so this is the one main thing that you can observe here, that at the top, this is the highest score. Means the first sequence here, it has aligned much better with our query sequence among these sequences. Okay, so here it has the highest score 616, and again highest the score, the better is the alignment. And here let's scroll down, here we have the score 377. So lower the score, the weaker is the alignment, but still all of these sequences, they have aligned very well. They are all a very significant match with our query sequence, because here it is 100%, E value is also low, or here percentage identity is also very good. Okay, now here we have accession number. So all of these sequences, these are the accession number of these sequences. So if you want to view these sequences, what you can do, you can just click on this accession number and it will open the sequence. For example, let's click on it. So this is the record of that sequence. You can either see GenBank file or either you can see the FASTA file. Let's open the FASTA file. So here is the FASTA file. Okay, so these are all the accession numbers of these sequences, and this is the accession length. Means it has 740. The size is 740 base pairs. So here let's go back to GenBank again. Here we can see it is 740 base pairs. Okay, so do we have any question in these sections? The maximum score, the total score is also 616. Query coverage, E value, percentage identity, accession length, and then the accession. So are we understanding these sequences, that they have aligned with our query sequence, all of these sequences? And here we can see the graphic summary. Now, here in this graphic summary, this is a visual representation of the BLAST results. All of these results that you are seeing here, it is the visual representation of all of these results. So instead of showing us a table, here BLAST displays them as graphically. Okay, so first of all, let's see this blue bar. We can say it is blue in color. So this blue bar, it is at the top, and query is also written here. So this blue bar here, it represents our query sequence. Okay, so from position number 1 till the position number 333. This is our query sequence. And all of these red lines that we are seeing here, so each red line here, it represents each BLAST hit. So do we understand the term hit now? That all of these are BLAST hits. Okay. So here each red line, it represents all of these sequences from here. Now you can see some colors here. Here we have black color, blue color, green, purple, red. So all of these lines, they have red color. Now why are all of these lines are in red color and not in blue, green and purple? So these lines, they are in red color, means they have, here we can see the number is greater than and equal to 200. So red here, it indicates a very high alignment score. So we can see the score is, the highest score is 616 and here the lowest score is 377. So here in the graphic summary, the color of these lines, means the color of all of these sequences are represented as this. Here it is representing the scores. So for example, if this sequence here, this red line is representing a sequence. So here, if this sequence, the score of this sequence is, for example, 90, so its color will be purple. Okay, so as we can see, all of the scores of all the sequences, they are greater than 200. So that is why they are all in red lines. Now, one more thing that you can observe here. Here you can see it is covering from starting till the end. This was the first alignment. And here as you see down here, we have some difference. Here we are seeing some difference and here we are seeing some difference. Now, here, let's see this first sequence here. So this first sequence, what it means, that it is from position 1 to position 333. It aligns completely with our query sequence. That is why we are seeing this long line. And here, it is a little shorter line, which means this sequence here, it matched only part of our query sequence. Okay, so here that is why we are not seeing the red line here. In the same way, we are seeing the red line is starting from here until here. Means these sequences only matched in our query sequence from this part till this part. So are we clear with the graphic summary? Do we understand the graphic summary? How to read the graphic summary? Okay, so graphic summary, it just provides a visual overview of the blast hits and it shows us that where each database sequence aligns with our query sequence and what is the strength of the alignment. Okay, so here you can see all the alignments. So here this was our first sequence. So here you can see our query and this is the subject 1. Here this is the second alignment, our query, and this is the another subject. So these are all the alignments here. Every single thing that is written here, every single sequence, every single alignment here, these are all the alignments that is showing to us. Okay, and here we can see the taxonomy as well. If you're interested in taxonomy, you can see the taxonomy as well. So do we have any questions related to BLAST? Do we understand the BLAST now, that how fast it is and how in a short amount of time it just aligned our query sequence with millions of sequences in the database? Okay, so here we discussed about the nucleotide BLAST. Let's go back. Here we have the protein BLAST as well. So everything is the same. Okay, from BLAST N to BLAST T, BLAST X, everything is same. Whichever BLAST program you will open, everything is same. You have to just paste your sequence and you have to click on BLAST. But in this protein BLAST, I want to show you something. As we have already discussed that to compare the protein sequences, it is a little complex, right? That is where we learned about the substitution matrices, correct? And in the substitution matrices, we learned about PAM and BLOSSOM. So here, if you see these algorithm parameters, you can see that the matrix it is using is BLOSSOM 62. And from here, you can select all of these substitution matrices. But BLAST by default already uses BLOSSOM 62. So there is no need for you to change these substitution matrices. Okay, so here the purpose of showing you this is to tell you that we learned about the alignment in the protein sequences. As there are different amino acids, there are chemically similar and they're chemically different as well. So that's why they need substitution matrices for a better alignment. That is where we learned about Pam and Blossom. So Pam was a little older, that is why Blossom was developed later. So BLAST uses Blossom 62 by default because it is in the middle. We learned about this number 62. It is in the middle. So it is not very lower, it is not very higher. So BLAST uses Blossom 62. Okay, so are we clear with this Blossom 62? Okay, so your assignment is that as we have performed the nucleotide blast, you have to perform blast X, T blast N, and protein blast as well. And you will share the screenshot of your answers in your assignment. Okay, I will share the assignment details in the Google Classroom. So you will perform the blast. You will not perform nucleotide blast as we have performed it. So you will perform all of these three blast programs and you will share the screenshot of the alignment. All the details of the assignment will be uploaded on the Google Classroom. Is it clear? Okay, so everyone raise your hands so that I can mark your attendance.